Welcome to Start Disrupting, podcast celebrating disruptors and game changers in science and technology. Recently, a dear and vital connection put me on to Antarctica Artists and Writers Collective. That got me really going down a trail of investigating, and I found myself reading an issue of Nautilus that highlighted one of my guests today. It really sums things up nicely in terms of if Antarctica had a voice, it would be Jim McClintock. And that's how the article opens up. And I found myself further in the article hearing stories of Dr. Bill Baker's work on identifying compounds for MRSA. And I said, what a fantastic opportunity to bring this story to the CRC and all those who are interested in the technologies. So let me start with a proper introduction for Jim. He is a endowed university professor of polar and marine biology at the University of Alabama in Birmingham. He has over 290 scientific publications, author of a fantastic book I highly recommend, Lost Antarctica, Adventures in a Disappearing Land. He's been a TED speaker, and notably, if you know the Moth Hour, it was a speaker and told a fantastic story of adventures in Antarctica. So Jim, welcome.
Great to be here, Brett.
Thanks. And Jim's colleague, longtime colleague of Exploring Antarctica, Bill Baker. He's currently professor of chemistry focusing on chemical ecology and specifically identification and cultivation of natural products at the University of South Florida. Bill did his work at University of Hawaii and studied a variety of marine and environments, and specifically working on secondary metabolites in marine life. And that's really where this story begins. And what I've really found interesting as well is his publication as the chief editor in Marine Biomedicine, From Beach to Bedside. And I just absolutely love that title. Bill, welcome to the podcast, and I understand you have a few special connections to Virginia Tech as well.
Absolutely, yeah. Thanks for inviting me, Brett. But yeah, my good friend Dave Kingston, he and I go way back in our primary professional organization, American Society of Pharmacognosy, is where I first met Dave. But one of my former undergraduates is this year a brand new assistant professor in the Department of Chemistry, Emily Meavers. I want to give her a little shout out because she's doing much of what she did in my lab, chemical ecology wise, and I think she's a young up and coming star in the field.
Fantastic. She's got a home here in Blacksburg, and we're looking forward to great things and helping her cultivate all the technology. So let me dive into this. Why are we talking about Antarctica, and where is this headed? We're really interested in the ideas of understanding how Antarctic polar ecosystems can really impact mankind. And so I want to start at the beginning. The most important thing, and for Jim, we really want to orient the listener and paint a picture. What I found most fascinating about your work is how you paint a picture. And most people really think of the cold and windy terrain, but this story of this work starts with a cold plunge below the surface. So Jim, let's drill a hole in the ice, strap on our tanks, and have you take us into your world. Paint a picture of what we see when we pop out under the ice.
Well, first of all, let me tell you, your heart is beating very rapidly. The first time I went down through that hole, I think the only term that I could use would be stunned. And as Bill knows very well, you drop below the ice. The ice is about 6 to 8 feet deep. And the thing that strikes you when you're diving below the ice at McMurdo Station, where this was done initially, our diving, is the visibility is just stunning. It's crystal clear under the water. And I think in my book, I use the term sort of you're floating down through a ceiling of blue light. Felt a little bit like I was in the Sistine Chapel, just looking at this tremendous scene. And on the seafloor, as far as you can see, is life. Sea stars and sponges and soft corals and giant Nemertean worms. And, you're sort of have this visceral sense above the ice that there's not going to be much down there, because above the ice, there's not much there. You don't see much in the way of biology, at least as far south as McMurdo. But below the ice, you have a diversity of organisms that's truly stunning.
That's amazing. And it's probably not something that someone would expect. Bill, was that when you saw that, and when you see it for the first time, what hits you, and how do you think about this from a biodiversity perspective, and frankly, an opportunity for discovery?
No, absolutely. In fact, the first time that Jim and I spoke about doing the kind of work that, right, I mean, I cut my teeth in Hawaii, right? And the biodiversity in Hawaii is great. Pretty much anywhere you jump in, you see sponges and corals and fishes, and right. And so first time I talked to Jim, and he says, do you want to do this kind of stuff in Antarctica? And I'm like, is there anything there? are there sponges and corals and tunicates and things? And Jim just kind of laughs and said, don't worry. And no, absolutely, the biodiversity. So the biodiversity is, of course, what's key to driving what is interesting for me, to driving the chemistry. If there was only one organism that lived on the seafloor, that organism wouldn't need chemistry. It's all its own kind. It doesn't need chemistry to interact. But as soon as two organisms start competing, chemistry is one of the things they can use as part of that competition to keep one another in check, to one poison the other so he gets the resources right. Yeah. You start adding more and more organisms into that, and chemistry becomes a very integral part of ecology.
Well, and I think, Bill, you're hitting the nail on the head, and Jim, you really tell that story well in terms of the shrimp and the butterfly and the backpack. Maybe that's a great short example of how chemical defenses can be co-opted, and what you see as opportunity there.
Oh, that was quite the day. I'll never forget seeing a little sea butterfly. It's a little snail with wings that have evolved, and it flutters around in the sea. And it was being carried by a little amphipod, a little crustacean. And I brought it up to a colleague, John Jansen, at the aquarium in McMurdo, and we looked at this puzzling relationship. And well, first of all, we wanted to know if it was happening commonly. So we sampled the sea there and found that there were a large number of them carrying little sea butterflies. Why would they go to the trouble? Why would a little crustacean grab a little sea butterfly and swim with it? It's difficult to catch. It's heavy to carry around. And to make a long story short, what we found is that there is a chemical in the sea butterfly that is very distasteful to predatory fish. And if you remove the little sea butterfly from the back of the amphipod and offer it to a fish, the fish will eat it happily. If you leave the sea butterfly on the back of the amphipod, the fish will take it in the mouth and then spit it right back out. Away goes a happy little amphipod carrying its chemically defended sea butterfly. And when we poked around in the literature, we discovered that this was, if not the first, one of the very first examples of one species abducting another species because of chemical defense. And that really helped us get it published in Nature, which is a very prestigious journal. So that was an exciting finding and an example of how complex these chemical relationships can be and how important they are.
Yeah, I think that story really talks a lot about the ecology and what a closed system somewhat it is, as defenses evolve in that environment.
And I should add that the chemistry in that sea butterfly was defined and described by one of Bill's postdoctoral fellows or graduate students, right, Bill?
Graduate student, yeah.
Yeah.
So, Bill, from beach to bedside, I love that because it assumes the bench is not the starting point. Typically, it's bench to bedside. You're saying the beach, which, I love. Give me an idea of how you approach that, and then how do you think about, and maybe a few examples of some of the more notable ones. And by the way, pharmaceutical research starts with natural products. So we know paclitaxel and a lot of natural products derived and ultimately cultivated through screening of plants and compounds and so forth. Tell me, we have companies here in the CRC that are working on MRSA, gram-negative bacterial infections, malaria vaccines, working on oncology and specifically melanoma. So tell me a little bit about how your discoveries have impacted some of those areas.
Sure. So yeah, the process, the tried and true process is what's known as bioassay guided purification, right? And this is where essentially us field workers, right? We go out into the field in Antarctica and Hawaii and wherever, you know, we happen to take an interest in, and we scuba dive and we look for sponges and corals and tunicates that look like they might be chemically rich, and that's, you know, it's you can't just look at a sponge and say, oh, that's going to be, you know, the next cure to cancer. But you see things, you see sponges that have grown together and have a dead zone in between them, you know, and there's some kind of competition going on there, right? And so that's sort of an indication that that might be a good sponge to take a look at. But at the end of the day, you end up collecting a lot of specimens and you screen them. So screening is a big part of our program. You take extracts of these things, and you know, just point out these chemical extracts. This is very simple stuff. You make a chemical extract every morning when you make a cup of coffee, right? You extract the chemicals out of that coffee bean and put them in your coffee. And if you were to evaporate the water out of your coffee, the chemistry that's left in the bottom of that cup is all the chemistry that was in the coffee bean. So you've used a solvent to separate the chemistry from the biological material, right? And that's exactly what we do when we put those chemical extracts into MRSA screens and cancer screens, and, you know, see if any of that chemistry is bioactive. And then we do a series of chemical manipulations on extracts that we find that are active to isolate the active component of that. And then from there, you know, we largely depend on biologists to take those forward, right? So once we've shown the biology community that this compound has activity against MRSA, you know, pharmacologists can start looking at the drug-like properties of the molecule. You can start looking at mechanisms of action and whatnot, all those developmental steps that are required to take something forward, like Taxol. Taxol was discovered the very same way. It was just a tree that was extracted instead of a sponge. But we've had a number of examples of marine organisms who have advanced. Bryostatin is probably one of the most famous from a bryozoan, but there's other tunicate compounds like tenosidins. There's a anti-cancer product from a tunicate that's been approved for clinical use. Cone shells, their protein toxins have been used as analgesics. So there's a number of things that have gone both from the sea. You know, you say that nature is, you know, the source of so many drugs. It's not just traditional. Even today, you know, most drugs have their either direct origin, a natural product from a sponge or a tree, but certainly inspiring chemists often to make derivatives and to build off parts of the molecule that they know have biological activity. And so natural products is very key to our modern medicine. Absolutely.
Yeah, and I think that discovery comes from inspiration and discovery comes from all sources. I was really intrigued recently, and one of the things that our younger entrepreneurs and investigators are seeing is the leveraging of synthesis, synthetic approaches, and frankly, bioinformatic driven and omics driven approaches. And so, you know we have several companies that are working on rationally designed peptides, for example, that can be inhibitory against certain negative genes for antibacterial properties. And one of our famous companies is Teculon doing that. And yet at the same time, I think this intersection of all of these technologies could most likely help accelerate what you're discovering and bring those into bedside faster. And I will give a shout out for one of your students, Nicole Avalon, who did a fantastic job of basically looking at a mechanism and then developing a strategy to help create the biosynthetic machinery. And I think as I unpack this question a little bit, you know, investors, entrepreneurs, people who want to take something forward into the clinic, I mean, you know this, it's a very expensive proposition. So how do you develop the understanding faster from discovery so that you can scale it up and get it into clinic without having, you know, decades to go through the further validation? So talk a little bit about the intersection of these technologies, omics, informatics, and how does that help accelerate what you're discovering from the natural world?
Yeah, Brad, I think you know, you really hit the nail on the head. I think everybody in my community right now is really driven to innovate in a manner that accelerates what we do. So what I described for you, you know, where you take these extracts and you screen them and you purify metabolites, this takes a long time. It takes too long. And so what we're doing is we're developing these bioinformatic approaches to accelerate the process. We have a big project right now with a group in Ireland, actually, where in the deep sea around Ireland there are corals. So it's a coral haven. It's beautiful. We send ROVs down and get high definition feedback up at the ship, and we see these beautiful scenes of just coral forests, right? And so one of the things that we want to do is rather than just, you know, collecting every coral and screening it in, you know, this slow process of biodiscovery, we want to see if we can use computational approaches to predict not only which of those corals is going to be active, but perhaps the georegion that we should go to find corals that have bioactivity. And so we have a collaborative approach with a group here at University of South Florida, doing essentially taking, you know, all known coral compounds and screening them against various targets in the protein databases. And that's going to focus us on perhaps species, perhaps genera of corals that are more likely to produce bioactive metabolites. But it's these kind of approaches that help accelerate the program, and that's the name of the game today, is trying to find those things before, well, for one thing, is with global warming is part of what we want to talk about today. Species are disappearing. We need to identify the ones that have the best potential and study them now.
Yeah, absolutely. And you hit the nail on the head, which is we've got this vast sort of compound library and opportunity ahead of us. We can't afford. We have one of our Virginia Tech researchers, Jeb Barrett, studies regularly at the Taylor Valley, and some of the stories of the winds that come off of that area. But he does a lot of work and studies movements of microorganisms that are tagged that you can identify from either erosion or changes in the soil. And he's picking up some remarkable findings. Jim, big picture. I mean, obviously Antarctica is the Earth's barometer. And we know what's happening here. Give us a sense of the pressure that's really on. And frankly, with warming and with these changes, what are we facing to lose here in the next 50 to 100 years?
So let me contextualize this with just reminding everybody what a gem Antarctica is in terms of its marine biodiversity. I mean, this is an ancient seafloor. It's the result of the separation of Antarctica from South America 40 million years ago. It really has had a very long time for evolutionary processes to diversify not only species, but chemistry. And so what a scary thing it would be to squander this resource. How is it changing? It's warming. Well, the area where Bill Baker and I and Chuck Amsler, our other colleague, have been working has increased about 10 degrees Fahrenheit midwinter air temperatures in the last 60 years. That makes it warming at the pace of the Arctic, if not even faster. And this is an 800-mile length of the Antarctic Peninsula. So what are you seeing? You're seeing melting of the ice, glaciers. You're seeing the ice sheets breaking up. You're seeing temperatures in the water warming, and the marine invertebrates that have lived in this water are very, what you call temperature sensitive. Many of them have not seen changes of temperature such as occurring, and we know that some of them are vulnerable to warming. And the other thing that people forget about with climate change is it's not just warming. We are putting CO2 into the atmosphere by burning fossil fuels, and a third of that is ending up in the world's oceans. And we have a process called ocean acidification going on. And the Antarctic, the Southern Ocean, is the canary in the coal mine for ocean acidification. It's simply the colder the water, the more CO2 is absorbed into it. So we have a situation, we've been doing studies looking at the effects of ocean acidification on marine invertebrates, and we're finding that some of them are vulnerable. Not all of them, but some of them are vulnerable. So we have a very rapidly changing system. We have some species that are already having problems, particularly invertebrates like the Adelie penguin, for example, is an example of a species that's really having a hard time and is disappearing as snowfall is increasing and covering the rookeries after they lay their eggs, and the eggs aren't surviving that. But so everything is at risk. And I think there is a sense of urgency. Another thing that we've found is that king crabs that live in the deep sea around Antarctica have begun to move up the Antarctic slope. Held at bay by low temperature on the slope for millennia, and now with warming, the Antarctic circumpolar current, the largest current on the planet, is beginning to warm. Those deep waters are warming, and for the first time we're finding billions of king crabs moving up the slope. And what does that mean? Well, it means if they get to the shelf, you have a marine invertebrate community that's never encountered a crushing predator, and we could have millions of them. And that means that those precious invertebrates that have yet to be looked at in terms of their drug potential could be lost. So there's many factors that are conspiring to make it important that we address the root problem of climate change, of course, but also do everything we can to expedite the process of looking for potential drugs in this system.
Yeah, that's well put. And, you know, to hammer home the point that you made, there's not enough time for species to adapt or evolve.
No, it's all about, yeah, that's the thing
these ecosystems have taken millions of years to evolve and stabilize and reach equilibrium. Yeah. So the process of adaptation or evolution is just, time just doesn't exist for that.
That's well said, Brett.
So I want to shift a little bit and give some of our listeners an idea of how can they get involved. So I'm thinking about two frames here. One is the young investigators really going into this area and really building on the work of giants like yourselves. How should a young investigator think about developing a research program and getting involved? And what kind of advice would you have for them? And either or both of you, feel free to jump in and answer.
Jim, why don't you go ahead?
Yeah, well, I do get interested students approaching me and about working in the marine biology of Antarctica, the chemical ecology, various other things, climate change. I encourage them to, get a good degree in biology at a good university. It doesn't have to be in marine biology, and then look for a good graduate program. And, you can kind of mine the National Science Foundation data banks and figure out which universities and which investigators are working in polar systems, and approaching one of those faculty that has an active program in Antarctica is a good thing to consider as you're looking at graduate programs. It's not entirely necessary that I didn't work with, necessarily go to work with my doctoral mentor because he worked in Antarctica and I got there, but it is a nice thing to keep in mind. Yeah, and go to the conferences where these kinds of studies and things are going on, meet people, don't be shy. Bill, you want to address the...
Actually, Bill, I'm going to jump in and ask you a slightly different question, the same question. In terms of, let's say, either entrepreneurs, investors, or interested parties who might want to take some of your discoveries and build on them, yeah, what kind of advice do you have for them? And are you seeing uptake and interest? And, how do we get more of the word out around the opportunity? Because my guess is for every one of your discoveries, well, you probably have 100 discoveries and maybe one makes it off the bench, so to speak.
Right.
Exactly. And so what I've done over the years is for the last 12 years, I've led a cruise to Antarctica for the public on climate change, related to climate change with Abercrombie and Kent, and it's fantastic. 200 guests from all around the world, largely American, but not exclusively. And inevitably, after I give my drug discovery talk aboard ship, I'll have 3 or 4 entrepreneurs come up to me and say, I've been in this drug discovery world for 20 years, and this sounds really enticing. How might I get involved? And so I say simply, Bill Baker. And I put them in touch with Bill, and I hope that something might come from this. But yeah, there does seem to be quite a bit of interest. There's kind of a buzz aboard the ship for a little while, talking about, well, gosh, this compound's really active against melanoma, and my aunt has melanoma, and what can I do? What can we do to move this along? And so that's been my avenue.
So, I'll say one of the real roadblocks to advancing the kinds of discoveries that we've made in Antarctica is supply. So you can't just, hop on a plane to Antarctica every weekend to go collect tunicates to beef up your supply, right? And so this is one of the things that is at the crux of what Nicole has been working on, to develop a biotechnological approach, essentially heterologous expression of the biosynthetic gene cluster that could supply sufficient quantities of a compound like this melanoma drug that Jim was talking about, so that there would be enough of it to study. And so one of the things that we see in my lab is one of the most important things moving forward is to try to develop these technologies for producing these materials. So many plants, Taxol, collecting Taxus brevifolia isn't that big a deal. And so Taxol is now a drug, right? Collecting palmerolide from an Antarctic tunicate is a huge hurdle. So, if we can't get over that, that's going to limit the developmental potential of that.
Well, there's opportunities for companies to just develop these pipelines, if you will, and develop a platform technology or the pipeline to be able to receive some kind of interesting discovery and really convert it and translate it in a meaningful time, because that's where the cost of drug development can be impacted and driven down. And so I think that's really highlights. And here at Virginia Tech, David Kingston is actually involved in the Center for Drug Discovery, and we actively look for similar collaborations. So I think there's a lot of work going on in the field. Absolutely. Jim, I'm curious because I know that the tourism industry somewhat has been ramping up, going to Antarctica. I think that's an interesting dynamic. How do you see that in terms of balancing potential impact and the traffic put on the ecosystem versus the opportunity to educate a global community? And what's your outlook on that?
Yeah, Brett, this is something I wrestled with for years. And to be quite honest, 12 years ago when I first started this, I did so with some trepidation. But, in retrospect, I think it's been very successful. The cruise companies that are working in Antarctica are very environmentally focused. They may have to reach a point where they actually have lotteries. They're going to have to think about the numbers of ships going down. But as it
Right.
stands today, you can spend 10 days, as we do on my cruise, traveling along some of the most beautiful areas of this planet and not see another ship the entire time. So it's not the Galapagos yet in terms of overcrowding.
Right.
And so, there's another issue. The carbon footprint of going on a cruise ship is not trivial. And so you have to balance that with the education that these people are getting about climate
Right.
issues. And I, the people on these cruises, some of them are very closely aligned with personal relationships with their congressmen, senators. And I have seen a number of things come out of them returning as Antarctic ambassadors to address big issues. So this has been very, very rewarding. I think that is, to me, is an important balance with the carbon footprint. And now I think the next big push in carbon issues is going to be the cruise ship industry. They're going to have to begin to regulate that, and I think they know it's coming.
Listen, I appreciate your time today. We're going to wrap it up, and Bill, thank you for your comments and input and advice on this. We're glad to have you, and Jim, it's just been an honor, and we appreciate both of you, literally both of you pioneers in really bringing this field into focus for everyone, and then really showing what the opportunities and highlights are.